So I confirm you, we are on air on YouTube.
So I think we can start at this point if everybody agrees. And I'm happy to introduce our today's speaker, John Hiscott, who is the director of the Laboratorio Pasteur at the Istituto Pasteur Fondazione Cenci Bolognetti in Rome, here in Italy. Dr. Hiscott is internationally recognized for his contributions to the field of antiviral innate immunity and host response to viral pathogens such as influenza, dengue, and chikungunya. Dr. Hiscott also studies human retroviral pathogenesis, and he is currently investigating the use of oncolytic viruses as novel immunotherapeutics for cancer. Prior to joining the Istituto Pasteur in Italy, Dr. Hiscott was the director of the Molecular Oncology Group at McGill University, Montreal, and the program director at the Vaccine and Gene Therapy Institute of Florida. And I can leave the words to John for his lecture.
Okay. Well, thank you very much. Good afternoon, everyone. As Giuseppe said, I'm here at the Pasteur Institute in Rome. And today what I want to talk to you about is some of the early events in innate immune signaling to the antiviral response. And I'm going to illustrate a number of signaling pathways and responses that the host makes to the early stages of virus infection. I think as we go along over the next few minutes, you'll see that there are a number of interconnections between what I'm going to talk about and the current epidemic. Well, I want to start today where I feel I have to start. This is a peaceful hillside park. The photo is taken from a historic wooden structure called the Yellow Crane Tower. And it's in a city that I have previously described as a beautiful city of 10 million people that nobody knows about, or few people have heard of. But sadly, as you can probably guess, the city is Wuhan. And I guess for time immemorial, it will be known for being the epicenter for the coronavirus outbreak. And pictures of deserted streets, people with masks, overcrowded emergency rooms seem to be the order of the day, not only in Wuhan, but now throughout the world. And I just want to say the last time that I was speaking, I pulled up a copy of the Johns Hopkins University live update. And at that time, which was end of February, as you can see, the center of the epidemic for COVID-19 was really centered over Hubei province and more broadly over China. But by that time, it had become clear that the virus was spreading in a pandemic form to Italy, I think with disastrous consequences I think we are all aware of. A couple of days ago, I pulled up the map, and the map has changed dramatically, as you undoubtedly know. All over Europe, the pandemic has spread. In fact, it's abated in China, and now the pandemic is really hitting North America, the United States very seriously. And in the United States, I think there was, or there has been, an incredible disorganized response to the virus infection. There has been almost, I would say it would be laughable if it wasn't so serious, a lack of leadership in terms of dealing with the pandemic. And what is really very striking is that there has been absolutely, not just in North America, but throughout the world, very little preparedness for a pandemic. That's a term that we hear a lot these days, pandemic preparedness. But unfortunately, I think that it's quite clear now that there needs to be a global rethinking of the whole issue of being prepared for these kinds of outbreaks of viruses. And I'll say at the outset, it's not as if we haven't been warned. In 2009, there was an epidemic or pandemic outbreak of H1N1. The original SARS outbreak in 2003, which was very much contained, but still was an indicator of what was to come. Zika, chikungunya, dengue, Ebola, of course, all of these outbreaks were contained through a very concerted effort on the part of countries and the World Health Organization. But it should have alerted, I think, world authorities that there was a palpable danger for an outbreak like this. So I just want to go briefly and talk a little bit about viruses, overview of virus infection and the kinds of diseases they cause. This is one of the most depressing slides. I can't believe that we survive anything here, but these are various diseases that are linked either directly or indirectly to virus infection. As you can probably guess, many virus infections gain access to the body through the airway, through the nose and mouth, or through the gut, and also a number of viruses that can cause skin infections. And there's quite a collection, as you can see, resulting in a wide range of illnesses and so on. My next slide is really an overview now of the kinetics of the immune response to virus infection. Just one second. And really, today what I want to focus on are very early events, the early triggering of the innate immune response to virus infection, the productions of interferons, often referred to as the first line of defense against infections. In subsequent lectures, I am sure that you are going to learn more about the innate lymphoid cells and NK cells and their activation, also early in infection, and the cytolytic properties and immune regulatory properties that they have as well. So these early events are, of course, part of a primordial system that protects against various pathogens. The adaptive immune system, particularly a generation of virus-specific cytotoxic T cells and B cells producing antibody against the pathogen, are all part of an adaptive response that, of course, is going to be ultimately central in clearing the virus. And in this schematic, you can see virus titer peaking early and then decreasing. Globally, what one can say is that the early events can often determine the extent of disease pathogenesis and or the response of the immune system. So I want to, so that's really where we're going to put some focus today with regard to the triggering of the antiviral response. So I want to give a credit to Charles Janeway, the late Charles Janeway of Yale University. It was more than 30 years ago, or actually about 30 years ago, that he initially proposed the pattern recognition theory, the idea that there was a primordial innate response to pathogens. The statement was, he contends that the immune system has evolved specifically to recognize and respond to infectious microorganisms. And this involves recognition not only of specific proteins, but also certain patterns common on infectious agents, but absent from the host. So certainly Charles Janeway for the work that he subsequently did on pattern recognition theory and innate immune activation. Had he lived, he would have shared the Nobel Prize in 2011 with the group Jules Hoffman, Bruce Butler and so on, who won the Nobel Prize for this work. 30 years later, now please don't pass out from the complexity of this slide, but 30 years later, there clearly has been a lot of work done in terms of understanding the patterns and understanding the signaling and understanding the activation of the antiviral response. And I will just point out, break it down a little bit. Of course, the toll-like receptors on the surface of virtually all cells are important components of innate immune response and pattern recognition. They recognize often bacterial cell wall components, lipopeptides, peptidoglycans, and so on. C-type lectins, another set of receptor molecules that will recognize often fungal-like molecules, and so on. In addition to the cell surface toll-like receptors, there are a subset of toll-like receptors that are associated with the endosome. So still in very close contact with the cytoplasm of the cell and the cell surface, but actually anchored within the endosomes. And these toll-like receptors, a subset of them, TLR3, TLR9, TLR8, and 7, recognize nucleic acids that, of course, are generated during the course of replication of viral pathogens. Over here are a set of sensors that recognize viral infection, either RNA sensors, and I will spend a lot of time discussing the Rigi signaling pathway, as well as a group of sensors that recognize DNA virus infection. And I will be spending a good deal of time talking about the CGAS and STING pathway. So these viral pathways also not only trigger an antiviral response, but also trigger the activation of inflammation within the infected cells. So these various groups of pattern recognition receptors then use a series of adapter molecules that are shown here and kinases, which ultimately feed down into a relatively restricted number of transcription factors. And those transcription factors are responsible for the initial activation of the interferons, pro-inflammatory cytokines, chemokines, and that's shown down here. The transcriptional regulators belong to the NF-kappa B family, the interferon regulatory factor family, as well as the AP1 related transcription factors. So from a very specific group of receptors through adapters and kinases, ultimately leading to the activation of transcription factors, then you move to the generation of a host response, which not only generates antiviral immunity, but also can trigger inflammation. This is a much simpler look at the TLRs and the innate immune response. For those of you who said, why didn't you just show this one? Well, maybe it's a little too simple. But here are the TLRs shown at the surface, several different TLRs, and then the collection of adapter molecules. In the case of the TLRs, the adapter molecule MyD88 is central to the downstream signaling. MyD88 associates with the C-terminal end of these adapters. And then through other adapters and kinases, again, as we say, the transcriptional activation mediated by NF-kappa B, AP1, and IRF3, IRF7 can be quite diverse. The types of readouts, so the types of responses that are generated, of course, innate immune responses, including phagocytosis, instruction to the adaptive immune response, cell-mediated immunity, antibody-mediated immunity. And in the case of extreme tissue injury or chronic activation of these pathways, septic shock, autoimmunity, and cell death. I just want to spend one moment about the issue of inflammasome activation. I'm going to use this slide here. This is the slide showing TLR4, which will recognize lipopolysaccharide, and then using MyD88 as an adapter, will activate the IKB kinase, which is responsible for phosphorylation and activation of NF-κB. Activation of NF-κB, binding to DNA, and gene activation then leads to the stimulation of a whole series of host genes, including inflammatory genes like tumor necrosis factor, interleukin-1, IL-6, and so on. It also increases the activation of components of inflammation. NLRP3, ASC, procaspase-1 are all components of the inflammasome. And in response to a second signal delivered to these cells, there is a strong assembly of an inflammasome structure, which actually can be visualized in various microscopy experiments as particular specks intracellularly. The activation of this NLRP3 inflammasome then leads to the production and release of interleukin-1 beta, interleukin-18, and other chemokines that might be contributing to cell death in cells that have been, that are triggering this response. And this is often occurring through IL-1 mediated pyroptosis. Boop, 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 boop. So my next slide is a stylistic schematic of the mechanisms of interferon induction. It's taken from a very nice review from a number of years ago by Charlie Rice and colleagues, where there was a detailed discussion of the interferon stimulated genes. Of course, as I've already said, there is an initial recognition of virus infection, really at the level largely on the basis of release of RNA into the cytosol or release of DNA into the cytosol, then leading to a signaling cascade which will activate the interferon factors, NF-κB, resulting in interferon production. The initial trigger of infection, the initial infected cell responds. It's something of an, I think it's something of an altruistic response, in that these cells will produce some interferon, which is then secreted into the surrounding tissue. And those cells will likely undergo a programmed suicide, apoptosis, and so on. So, but the release of interferon is one of the key events in the amplification of the innate immune response. Interferons will associate with the interferon alpha receptors, either IFNAR1 or IFNAR2, or it's a heterodimeric structure. And through the recruitment of a specific Janus-associated kinases, JAK kinases, so-called JAK kinases, then phosphorylation of the STAT proteins, STAT1, STAT2, recruitment of one of the members of the interferon regulatory factor family, IRF9. You end up now with this phosphorylation event leading to nuclear translocation and binding to an ISRE element, which is an interferon stimulated response element. So this is the element that actually stimulates the production of interferon stimulated genes. Those genes actually represent hundreds, several hundred, perhaps even 1,000 or so genes that have a variety of effects on the cells and tissues close to the infection. They include the activation of a number of antiviral effectors that interfere with virus multiplication at specific locations in the virus life cycle. It also includes negative regulators, so proteins that actually act back on the whole interferon cascade to downregulate the expression of the signaling network. And then there are also a number of positive regulators that I will come back to that actually play a role in amplifying the whole antiviral response to infection. So this paradigm gives, this figure gives a sort of a nice flow from the initial signal through to the activation of the JAK-STAT pathway, the release of the antiviral mediators that play a role in controlling virus multiplication. So what are these? So several parts of the virus life cycle, spoken very generally now, are in fact inhibited by the interferon stimulated genes. So factors like IFN M1, M3, they can inhibit the entry of virus. There are other regulators that will modulate the ability of the host cell to translate mRNA. A number of regulators that will interfere with RNA virus replication, and as such then limit the level of replication, as well as interferon stimulated genes that will affect assembly of proteins and ultimately release of virions from the infected cell. So given that there are hundreds of antiviral proteins being activated, given that they can inhibit at multiple levels of the of the virus replication response, problem solved. That we should not have any virus infections. We should not be suffering from pathogenesis due to any virus infections. But, and this is the point where I have for a number of years said that viruses are extremely good immunologists. They have been investigating the immune system much longer than we have. In fact, probably if you were to put together all of the virologists, immunologists, and infectious disease specialists in the world in history, it wouldn't come close to the millions of years that viruses have been investigating the early innate and late adaptive responses to viruses and finding ways to interfere. This is just a very limited cross-section of the JAK-STAT pathway. This is again showing the interferon receptors, the JAK kinases and TIC kinases that are part of the C-terminal end of IFNAR, leading to the activation of STATs and the stimulation of the interferon stimulated genes. Any of these circles in red are in fact viral proteins that act at different stages of these pathways to interfere with the early production of interferons. I think probably because many different classes of viruses have judged that this interferon signaling pathway is very crucial to the response of the host to infection. So the strength of this response is being balanced against the capacity of innate immunity to respond to virus infection. And that's why we often hear about the viral wars between viruses and the immune response. Clearly, there are literally hundreds of viral products that either use most of their capacity to interfere with signaling or actually only use an insignificant, or use actually the viral components in multi-component viral products to kind of interfere. So I'm just going to stop for a moment. This is one of a couple of stopping points that I'll make to see if there's anybody that wants to ask a specific question or a general question, either about pathogenesis, the experience that we're going through now with COVID, or in relation to the early immune response to virus infection. And
so guys, don't be shy. This is your turn to make questions. You can also use the chat.
Okay, don't be distracted by the pictures. There's no doubt about it that this very beautiful but haunted city is amenable to many different photographs these days. So, okay.
Okay, so we have a question by Francesco Castagna. Francesco, can you please unmute yourself and ask a question?
Sure. Thank you for. Yeah, my question is: are only innate immune cells able to mount an interferon response, or other cells of the body? Thank you.
No, that's a good question. In fact, I think most cells of the body have the potential to respond to virus infection. I think that, as you might imagine, the response of epithelial cells, fibroblastic cells, can often be quite different than, for example, dendritic cells or other cell types. But yes, I think most of the cells of the body are able to mount some response to virus infection. And I think that you saw perhaps indirectly reflected in that first slide, where there were so many different kinds of viral pathogens gaining access to the body. Certainly, for example, in the lung, epithelial cells in the lung, or even the myeloid cells in the lung can independently respond to virus infection. So there is a range of cells that respond, yes.
Thank you.
Okay, next question is from Ludovica Timperi.
Hi, can you hear me well?
I can, yes.
Okay, my question is quite general. So I would like to know, in your point of view, which could be the impact of the microbiota on the viruses? And if we have an impact of the human microbiota, like, I don't know, for instance, in the skin.
Yeah, that's a very active area of research. And yes, I think that there is a significant impact there. Perhaps maybe one of the easy ways to describe it is that, you know, in the cases of influenza, where you often have a infection in the lungs and a bad cough, the breakdown of some of the tissues, particularly the epithelial tissues in the lung, can lead to secondary infections with other pathogenic bacteria. So the rupture of the epithelial layers then opens up to the subsequent bacterial infection. But in general, the microbiota, I think, can have both a very positive and a very negative effect on the overall effect of viruses. This is an area that is being studied very significantly at this point.
Okay, thank you.
Thank you. Next question is by Natalia Torianova. Please, I would like to ask to the students to turn on their video if you can, and also to tell the university you're from. Thank you.
Hello, I'm from Verona University.
Hello.
I have a question. If you could probably comment on the ability of some viruses like herpes virus, and also probably of this new coronavirus, to persist in nervous system, in the neurons.
Yes, good question. In fact, you know, several viruses have a propensity to survive in the neurons, or if not surviving in the neurons, to subsequent to primary infection cause significant disease in neuronal tissue. Measles is a good one. Measles can be controlled largely through immune responses, and yet in very rare cases, persistence of measles can then lead to a very serious neurological disorder. In regard to the coronaviruses, I don't know specifically whether there has been any demonstration of persistence of coronavirus in neuronal tissue or not. So far, I think not.
Okay, thank you.
Yeah.
Thank you. Question by Agnese Rumolo.
Hi, can you hear me?
I can hear you.
Hi, I'm from the University of Pavia, and my question is, have you got any ideas on how SARS-CoV-2 escapes the innate immune response?
Well, when I'm not giving lectures online, I'm writing grants so that I can investigate that very question. But by analogy with the SARS, the first SARS coronavirus, it was considered to be extremely immunosuppressive, actually, in terms of the innate immune response. Specific genes, including the N protein of the virus, have been shown to have a very strong capacity to inhibit the early innate immune response. Coronaviruses are relatively large viruses, approximately 30 kilodaltons worth of DNA, and there is capacity there for these viruses to actually specifically produce proteins that can inhibit innate immune responses. So I think at this point, certainly the M protein, the N protein, and some of the non-structural proteins are implicated as being very strong inhibitors of innate immunity. So that early immune response, clearly we would want to have a very strong initial immune response. In contrast, the virus in initiating an infection wants to suppress that infection as much as possible. So yes, I mean, there are, by analogy with SARS coronavirus 1, there are a number of specific proteins that will interfere. I should say that at the end of the class, I'm going to forward a couple of review articles to you. One is coming from Alessandra Zavini, who is one of the members of my research group here. And she's going to talk about innate immune signaling. And the other review article that I liked very much was, has just come out on innate immune evasion by RNA viruses. And it's by Marjolein Kikkert, who is in the Leiden University Medical Center. And she speaks very eloquently about this interplay between viruses, virus proteins, and inhibition of innate immunity. And so I'll give you those. They will give you much more idea about the specifics of the proteins that interfere with innate immunity.
Thank you very much.
Okay, so we have more questions, if we may. One is from Lorenzo Cuollo. Lorenzo, please. Lorenzo, can you hear us?
Yes, now can you hear me?
Yes, there you are.
Yes, thank you very much. My question is about pyroptosis, which I know is a kind of controversial subject, but my question was, is pyroptosis a common cellular fate after the initial viral infection? And also I was wondering if when cytotoxic lymphocytes or natural killer cells kills the virus infected cells, is it always the response apoptosis, or it may also be sort of inflammatory self-destruction? Thank you.
Yeah. So yeah, I think I haven't not stressed that as much as I should, is that one of the consequences of activation of some of these signaling pathways is that it also can lead to cell death, either the classic apoptosis or an interleukin-1 related pyroptosis. And sometimes, and I agree with you, it's somewhat controversial. Sometimes it's a little bit difficult to distinguish the different mechanisms of cell death. In terms of cytotoxic lymphocyte targeting and killing of infected cells, generally, I think this can be both mechanisms, apoptosis or a pyroptotic mechanism that can lead to cell killing, mediated through intermediates like granzyme and perforin. So yes, I mean, there really is a capacity of triggering of innate immunity to kill the infected cells through apoptosis and pyroptosis. I will come back to that in a while on this subject.
Okay, thank you very much.
Thank you.
Thank you. Next question by Eleonora Sala.
Can you hear me?
Yes.
I have a question. In your opinion, is more important the timing or the magnitude of type 1 interferon upon viral infection?
They both are critical. The timing of the response and the magnitude of the response, I think, are two critical components of this response. Timing of the response, I'm trying to think what would be a good example here. If one were to look at Ebola, for example, I think that by the time the immune response or one of the other hemorrhagic fever type viruses, by the time the immune response is triggered, it's already too late. The virus has replicated so rapidly that the antiviral response is largely ineffective. The magnitude of the response, as well, as you can imagine, is going to be critical. If you inhibit very strongly early innate immune response, it may be too weak to completely inhibit virus multiplication, and therefore the virus is able to spread rapidly. A very strong response, ideally you would want to have a very strong response, but you also want to have a response that is limited in terms of time. That is, that there would be a shut-off mechanisms that would restrict the activity of interferons or other cytokines, simply because these are very powerful. If they are being produced constitutively or on a chronic basis, they can, of course, lead to autoimmune problems and so on. So yes, yes, you hit the nail on the head with that one. Timing and magnitude, I think, are both critical components.
Okay, thank you.
Okay, so thank you. We now have a couple of questions, if we may, from our YouTube chat. So I'd like to take the occasion to remind people that are following from YouTube if they can post their questions also on our Twitter account, because we're following that one, and so we don't miss any question. So the first question is by Alfredo Marchetti. He asks, is there a molecule able to directly target the interferon responsive element on DNA to bypass the molecular arms race with the viruses?
Directly bind to the interferon response element. Yes, there have been demonstrations of molecules that will bind to the ISREs. Sometimes, rather than being a partnered sort of dimer protein, it can be a single molecule that will bind and basically occlude the activation of the ISRE. That is, I think that is not such a common mechanism, but yes, there are examples. I'll mention one later. For example, one of the antioxidant response elements has been shown to bind, to the promoter elements in interleukin-1 and suppress expression like that. But I don't think there is a global inhibitor of ISRE activity.
Thank you. Alfredo is a student from University of Milan, and the next question is from Alex, another student from University of Milan. Is there, into the immune cells, an alternative pathway used to sustain the expression of the antiviral genes? Alex Pezzotta.
Sorry, an alternate pathway to sustain expression?
The expression of the antiviral genes.
Yeah, not that I know of. No, I mean, I think the answer is there are several different ways to actually activate the antiviral response. I've shown illustrated a couple of those, but there are more indirect ways that it can be activated. But sustained, no. To sustain that response is potentially dangerous for the cell.
Okay, and then we have one final question in our chat. Is from Maria Nele Romeo. Please, Maria, unmute your microphone.
Hi, do you hear me?
Yes.
I'm from Sapienza. I have a question about SARS-CoV-2 infection.
Dicono ancora una volta, can you hear me?
Okay. Okay. In SARS-CoV-2 infection, there are some people that are asymptomatic and other that have a very strong inflammatory response. Do you think
Please, somebody should unmute their microphone. Anna Pasotti, I think.
Hey, I'm here.
Okay, Maria, can you just repeat the question? I got some interference in there.
Yes. In SARS-CoV-2 infection, there are some people that have asymptomatic response and others that have a very strong inflammatory response.
Yes.
Do you think that immune response can be the difference in that case?
Yes, I think that look, these are the very central questions that we're trying to understand, that in some individuals, probably just a strong immune response, strong innate immune response allows these people to control virus infection. On the other hand, in the case of COVID-19 disease, there seems to be a mechanism or multiple mechanisms not understood that basically represents some sort of a switch in which now, rather than having a virus infection that is chronic, somewhat controlled by immunity, the switch is such that you then move to a massive cytokine storm, a massive release of multiple cytokines and chemokines, and that cytokine storm is part of an immunopathogenic mechanism that leads to severe disease.
Okay.
And it probably, when you move to this very severe cytokine storm, in the case of COVID-19, pneumonia, and so on, it may not be directly due to the virus anymore. It's more the uncontrolled immunopathogenic response of the host. But it's not at all clear, immunological, virological, whether it's genetic components as well, that are contributing to the very severe disease in these cases.
Okay, thank you for the response.
Okay.
Thank you for all these questions, and thank you for answering, John. We can go to the second part of
Thank you for the questions. Very good questions, everyone. Okay, so I'm going to go through the cytosolic Rigi receptor pathway, and as the title says, it's a cytosolic pathway. It's recognizing RNA virus infection, and I like to think of it as the initial trigger of the antiviral immune response and of the whole immune response. These are key interactions that determine interferon production and then subsequent activation, mobilization of the immune response. So with Rigi, Rigi is a cytosolic helicase Virus infection, an RNA virus infection, such as influenza or coronavirus infection, ultimately has to release its RNA from the viral coat in order to replicate. That's, of course, it's the main purpose here. The binding of RNA by Rigi is very specific. It changes the conformation of the molecule of Rig and actually exposes what I'm showing here as 3 little circles. It exposes protein-protein interaction domains, and the Rigi then is able to interface with another molecule, which is called MAVS. MAVS is, or IPS, it's got a number of different names depending on which group isolated originally. But this protein is on the outer membrane of the mitochondria, and it actually acts as a signaling platform, and it recruits the adapter molecules that then lead to the activation of a couple of different pathways. One is the activation of kinases. These are related to the I-kappa-B kinases. They lead to the phosphorylation of IRF3 and IRF7. The phosphorylation of these proteins then leads to nuclear localization. And also, it also has recruited kinases and adapters corresponding to the classical IKK pathway. This leads to the release of NF-kappa-B, P65, and P50. And then down here, you go back to this mechanism of activation of inflammatory cytokines such as TNF and IL-6, as well as the activation, using the other arm of this pathway, the activation of interferon gene regulation. So I've been working in this area for a while, starting from the nucleus out, but I like to describe this pathway in terms of specificity, complexity, and diversity. And I will kind of illustrate what I mean by that in a second. But it encompasses, I think, in many ways, many of these signaling pathways that respond to virus infection. In terms of specificity, I won't go too much into the structure of Rigi, but I've mentioned that there are protein-protein interaction domains called card domains. The protein itself is an RNA helicase. That's the region in the middle of the protein, which is involved in unwinding RNA structure. And then there is it was called a repression domain. It's more of I think it would be considered an activation domain at this point for Rigi. And Rigi recognizes RNA that contains a 5' triphosphate structure. That's a structure that's generated on the 5' end of viral RNAs as they are beginning to replicate intracellularly. So that becomes the pattern recognition moiety, if you will, that allows the specificity of Rigi to interact with viral RNA. MDA5 is another member of this pathway. It does not have a specificity for 5' triphosphate RNA, but rather has a specificity for long double-stranded RNA, which is generated by many viruses during replication. Poliovirus comes to mind as one of those that generates a long double-strand RNA that is recognized by MDA5. This is a structure of the C-terminal end of Rigi. As you can see, there's a hydrophilic pocket in the C-terminal end of this protein. And it is this hydrophilic pocket that actually forms the recognition domain for this 5' triphosphate group. And that specificity then results in the conformational change in Rigi such that it now reveals these card domain proteins that then are able to interact with the MAVS protein. So that provides quite a specific interaction involved in the initial recognition of incoming viral RNA. Okay, so complexity. I mentioned already the MAVS protein. Let's see, mitochondrial antiviral stimulator, I think is the way it was originally described. But as I said, there were several alternative numbers. So this was an interesting protein when it was initially identified, because it was a protein found associated with an outer mitochondrial membrane, and yet the association with the mitochondrial membrane was playing a key role in triggering antiviral immunity. Subsequently, a number of groups, particularly the group of James Chen in Texas, demonstrated that in response to Rigi signaling, that this actually MAVS protein generates a rather large aggregate, not unlike a prion-like structure, shown schematically here on the cover of Cell, and shown here schematically that in response to Rigi response to double-strand RNA, there is the association of Rigi with the outer membrane portion of MAVS. And then both the Rigi molecule and the MAVS molecule, they polymerize, they form relatively large aggregates. And these are then part of the scaffolding mechanism, if you will, that recruits the kinase, the adapters, and ultimately the transcriptional proteins that work downstream to turn on antiviral genes. Okay, one example that I would like to show. 1 second here, I've got some. There we go. Okay, one thing I would just like to point out is with regard to one of the transcription factors, IRF3. It's a personal favorite of mine. The protein structurally has an N-terminal DNA binding domain. That is the portion of the molecule that actually interacts with DNA. There is a long domain that's called an IRF association domain. This is the portion of the molecule that allows protein-protein interactions. And then down here at the C-terminal end is a regulatory domain. The regulatory domain of IRF3 has a number of key serine and threonine residues, and those serine threonine residues actually play a role in the activation of IRF3. And okay, this is a Western blot that illustrates a couple of points. First of all, IRF3 sits in the cytoplasm of resting cells. IRF3 is constitutively present, and it is in most cell types. In response to virus infection, but not interferon treatment, IRF3 becomes phosphorylated on these C-terminal residues, with a real focus here on the serine at 396 and 398. The next thing that you can see is that in response to virus infection and phosphorylation, IRF3 actually moves to the nucleus where it is performing a transcriptional function. Other studies, just see if I can get this to, there we go. Other studies looking at the details of IRF3 activation show that the single event of phosphorylation of these residues actually results in the protein becoming a dimer. It exposes a nuclear localization sequence, which is actually, I think it's in this region, proline-rich region. It also allows the association of a co-activator, CBP300. And then finally, the transactivation of a limited number of target genes, one of which is interferon beta. RANTES also been shown to be regulated through IRF3 and interleukin 15. The close cousin of IRF3 is IRF7. You will see that it also plays a key role in activating interferon response genes downstream of interferon alpha signaling. And IRF3 has, in general, a similar mechanism, a C-terminal activation sequence, and a number of regions that play a role in the activation of downstream antiviral genes. One quick structural ribbon diagram here. This is the C-terminal end of IRF3 based on crystal structure. And it's referred to as a very closed, condensed autoinhibitory structure in the resting state. After phosphorylation, these are the sites of the phosphorylation that takes place at the C-terminal end. And as you can see, that phosphorylation is at least in part exposed and responsible for the conformation change in IRF3. So an inhibitory domain of the protein is released, and actually this becomes the domain where the transactivator CBP300 can actually physically associate with IRF3. And this structural analysis came from a structural biologist lab and was very satisfying for us because it really confirmed much of the biochemical work that we had done with regard to activation of IRF3 and IRF7. And so in terms of diversity, the third part of this, so we have specificity with regard to the interaction of Rigi with 5' triphosphate RNA, a complex interaction and generation of a prion-like structure and aggregate by MAVs and other words, and diversity in terms of the types of genes that are being activated. This is a waterfall plot. It analyzes all the transcripts in the cell using an array procedure. And as you can see, very rapidly after you treat cells with 5' triphosphate RNA, you generate a large number of genes that are being activated, as well as a number of genes that are being repressed in response to this triggering of the 5' of the Rigi pathway by 5' triphosphate. Most of the genes that are being activated by the pathway are many of the antiviral interferon stimulated genes that I mentioned right at the beginning. And then there are almost as many genes that are being repressed. Having been someone who was interested in antiviral gene regulation, I spent most of my time paying attention to the genes that are activated. In fact, it's the bioinformaticians that pointed out that you've got as many genes shutting down as you've got activated. These probably are reflecting important functions as well. But here you can see now, if you do an analysis of the genes, they involve interferon signaling, they involve actually TNF signaling, activation by IRFs and so on. And these are largely the triggering of the antiviral genes. If you now group this in a very different way, looking at closely related patterns of genes, you can find a number of closely related genes being clustered together as part of either the activation or the repression of gene networks. As you probably can guess by now, you see the activation of STAT signaling, NF-kappa B regulation, ubiquitination elements are also upregulated because ubiquitination plays a key role in controlling the pathway. But at the same time, there are a number of specific networks that are being knocked down or rather inhibited as a consequence of infection. And many of these fell into the category involved in cell cycle regulation. So cell cycling seems to shut off relatively quickly. There are changes in metabolism, dynamics, and so on, at the same time that the interferon genes are being activated. For those of you who have a favorite gene or a favorite pathway, you can probably imagine that there are hundreds of different genes here that could be analyzed and could keep you busy for years or decades. I'm going to focus on one of them. You probably can't see what its name is, but it's called TMEM173. It's one of the genes that's being upregulated by interferon treatment and by Rigi. And TMEM173 is also known as STING, stimulator of interferon genes, I think was the original name for it. What is STING? Well, STING turns out to be, I think, an extremely important regulator and sensor of intracellular foreign DNA. So STING actually sits on the endoplasmic reticulum and plays a key role in sensing not RNA entry into the cytoplasm, but DNA in the cytoplasm. It then, as a consequence of the interaction with DNA, it will also dimerize and recruit those kinases, the TBK kinase, the IKK kinase, that lead to activation of NF-kappa B and IRF3. And there's IRF3 again, going into the nucleus and turning on gene regulation. On the right-hand side here is a ribbon diagram of STING. And as you can see, it sits as a dimer, almost like an open hands, if you will. And in the very center between the two monomers, there is the association of a cyclic dinucleotide. And this turns out to be a key activator of the pathway. So it anchors itself into the endoplasmic reticulum. It's forming a dimer. And then once this molecule GAMP binds to it, it actually changes conformation and oligomerizes. And that's, again, a common theme in terms of the antiviral response. So this is a schematic of more detail of the STING pathway. The virus here is a DNA virus. It releases its DNA into the cytosol of the cell, probably during the course of uncoding or early transcription. And that DNA is sensed not by STING directly, but it's sensed by an enzymatic activity called the guanosine monophosphate adenosine monophosphate synthase. And what or abbreviated to cGAS. And what cGAS does, it actually binds the double-stranded DNA that is in the cytoplasm. So this is the proximal sensor of foreign DNA. It then uses a GTP ATP to generate a molecule that I just mentioned, GAMP, which is a GA monophosphate. But it's not a usual phosphate. It's linked by a 2', 3' linkage rather than the usual 3' to 5' linkage. And this cGAMP is a second messenger that then binds into the interface between the two monomers of STING, changes conformation. You then recruit again those kinases, activation of NF-kappa B and IRF3. So you actually now, for DNA, have a similar parallel pathway for the response to a DNA virus infection, such as a herpes virus infection. What was also discovered, and I will just briefly mention this, is that this mechanism of activation is very reminiscent of the mechanism of activation of one of the interferon stimulated genes, oligoadenylate synthase. It's an enzymatic activity that actually recognizes RNA and then uses ATP to generate an oligoadenylate, again, linked by 2' to 5'. So this oligoadenylate, 2', 5' oligoadenylate is again a second messenger in this pathway. And what it does is it's not going to interact just randomly. It interacts with a specific RNase enzyme, RNase L, activates RNase L, and that RNase is capable of degrading RNA structures, double-strand RNA structures, or single-strand RNA with a double-strand secondary structure. So you have, in fact, a pathway, two more or less parallel pathways. One, this cGAS STING pathway, which can recognize and respond to a DNA virus infection, and the Rigi pathway and the associated antiviral genes, including oligoadenylate synthase, which can then lead to specific degradation of an RNA virus infection. Okay, so viral DNA, bacterial DNA, mitochondrial DNA in the cytosol. This is kind of a more elaborate schematic of what I've talked about. But what has become clear over the last few years is that it's not just responding to viral DNA, but it also is recognizing DNA that gets into the cytosol in an inappropriate manner. You can imagine most DNA is supposed to be packed away in the nucleus. And as a consequence of infections or DNA damaging agents or stress situations, DNA can make its way into the cytosol, and that can be recognized by the cGAS STING pathway. And one of the more common types of DNA that actually get released into the cytosol is mitochondrial DNA. So when the cell is damaged, when mitochondria lose integrity, mitochondrial DNA can often be released into the cytosol. It then is recognized by cGAS, and the whole pathway recapitulates itself. And in this case, results not in triggering an antiviral response, but also triggering the death of the cell. So mitochondrial DNA inappropriately found in the cytosol can lead to the activation of the cGAS STING pathway. I'm just going to stop. Okay, it's springtime. It's time for STING fever. Let me just comment on this for a second. So you can guess that if you have a small second messenger RNA that can stimulate cGAS STING and potently trigger antiviral responses, that this might be a good therapeutic target. And in fact, STING has become a real darling of the pharmaceutical industry. I've listed just a couple of titles here from very recent articles. And I think you can see STING activation will stimulate immunogenic cell death and enhance anti-tumor immunity in neuroblastoma. That's an experimental study showing that STING, when you stimulate STING, you can also trigger cell death in tumor cells. Another review on STING pathway as a cancer therapeutic. Again, here, an argument being made that STING actually can complement immune checkpoint inhibitor therapy, cancer immunotherapy, and augment the response. One study, I think one of the earlier studies, I think they described this as STINGvax, meaning it was STING vaccine. The group generated a cyclic dinucleotide that was different from GAMP, and it could also lead to cell death in different tumor models. On the other hand, I found an article which was just from the beginning of January this year saying drug developers switch gears to inhibit STING. Okay, on the one hand, we're talking about STING being used as a stimulator of anti-cancer therapy. On the other hand, we're now talking about inhibition of STING. You can imagine that STING is a very powerful regulator of the innate antiviral response. If it is not being appropriately regulated and is constitutively or chronically stimulating the antiviral response, this can lead to serious problems in relation to innate immunity and in terms of triggering chronic inflammation in cells that are being affected by STING. So there is both a positive, essentially an activator and a repressor strategies that are being pursued in order to analyze how STING is playing a role in enhancing cancer therapy or repressing inflammation and antiviral response. Actually a very good target for therapeutic development. Okay, I want to come back just briefly to the question of viral evasion by viruses. This is an article that I think appeared last year in Cell from an old colleague of mine, Adolfo Garcia Sastre, who broke down the ability of viruses to evade the antiviral response into different categories. And in a sense, we've kind of touched on a little of, on some of these along the way. There are some viruses that, most viruses in fact, try to protect their viral genome so that it remains intact, so that it can be replicated. And in some cases, actually structures that will be generated that could do this. Inhibiting interactions of the interferon response, I think you saw some of that. Regulating phosphorylation events. Some of the evasion mechanisms involve a transcriptional shutoff, RNA processing, and so on. So these strategies, multiple strategies are identified that can interfere with the antiviral response. This is back to the Rigi pathway. I'm not going to emphasize it too much other than to say here, through here is the Rigi pathway, and here again are different proteins from different groups of viruses that are able to interfere with this pathway. And they interfere at the level of specificity, as I've discussed. They interfere at the level of complexity of the protein-protein associations, and they interfere, in fact, with the diversity, some of the activation events by the IRFs and NF-kappa B. This one is a rather interesting one. NS1 of flu, it's a non-structural protein, 1. If you delete NS1 from influenza genome, it actually becomes a really lousy virus. It doesn't really cause much disease. And in fact, there are studies that are looking at this as a potential vaccine because it is so weak as a virus. And what NS1 does, it actually interferes with a ubiquitination event in Rigi. So even if RNA binds to Rigi, there's another event that's required, a ubiquitination event, before it can interact with the MAVS adapter called IPS here. So this NS1, very crafty, in fact, in terms of being able to target a critical event required for activation of NS1. And one can go down the list here and likewise identify rather ingenious strategies to interfere with antiviral response. The same thing is true with STING in that you can have specific points in the activation of STING being inhibited. Here you can see that SARS coronaviruses, hepatitis B virus, they actually will target STING and prevent a ubiquitination event. Dengue virus will actually cleave STING. Human papillomavirus will bind and inactivate STING and so on. So you can see again a cascade of events that interfere with STING triggering of the antiviral response. One thing about our lockdown is it makes for some very dramatic photographs. This is probably one of my favorite buildings in the world. It's rather haunting to see it like this. This is a stop point, so I just want to stop and take some questions.
Guys, second round of questions. Okay.
Second and last round, I think.
Oh, last one, okay.
Not sure.
So we already have a couple of questions. So from Federica,
Yes.
you can speak up. Federica?
Yes, I'm here.
Hi, there you are.
Hi, thanks for a really nice talk.
I'm not done. Oh, we're still going.
It's really interesting, so I'm
ordering
really happy.
Fine,
and for me. So I maybe missed it, but on which cell type especially do you study the transcriptome changes due to the Rigi?
Okay. Yes.
And do you think there is one cell type which is more important in this type of pathway, or all immune system is reacting?
So we have analyzed both human lung epithelial cells as an epithelial cell model. We've used fibroblasts; those are standard, fibroblasts in cell culture, and we have also analyzed dendritic cells, primary dendritic cells that have been differentiated in vitro, and characterize these signaling pathways in response to different viruses or using synthetic RNA. Are there differences in the pathways? Yes, there are a number of differences in the pathways. I think in the case of epithelial cells, fibroblastic cells, it's kind of a, if you will, a classic activation mechanism that I've shown. There is much more flexibility in terms of the activation mechanisms in myeloid cells and macrophages. For example, in response to some stimuli, you don't really use IRF3 very much because there's a direct phosphorylation and activation of IRF7. And so in these dendritic cells, IRF7 seems to be a real important regulator of the immediate activation. And myeloid cells, and also the other major cell type that is a major producer, of course, is the plasmacytoid dendritic cells. And you can imagine these are located in tissues that are, and their function is to be sentinels to response to foreign pathogens. And these cells respond very rapidly. Plasmacytoid dendritic cells, monocyte-derived DCs, macrophages, and so on, they will respond very rapidly to the pathogen. Cells like epithelial cells and fibroblastic cells, I would say they respond in more of a stepwise fashion to the response. So yes, that's what those are the systems that we've worked in regard to the activation networks.
Thank you very much.
Lorenzo Cuollo has a question.
Yes, it's me again from Sapienza University of Rome. Yes, I had another question, because you showed a Western blot in which was clear, at least in your model, that in order to activate IRF3, there needs to be an infection, and the stimulation with alpha interferon alone, it is not sufficient.
No.
But my question was, at least in this model, is it sufficient, the stimulation with cGAMP for activating IRF3?
Is cGAMP sufficient to activate? Yes, it is. It activates through the STING pathway. And in the next part of this story, I will come back to that question a little bit more. But yes, cGAMP can directly stimulate through STING and activate IRF3 and NF-kappa B, just as putting in 5' triphosphate RNA will activate the Rigi pathway directly. The activation mechanism with a virus is a little bit slower, and as you can imagine, the virus has to enter the cell, uncoat, release RNA, and then that RNA would be sensed, for example. So the kinetics of a virus activation of those pathways is a little bit more delayed. So in one case, you're looking at a response in 1 hour. In another case, you're looking at the response over 6 hours. So it's still pretty rapid.
Okay, thank you very much.
Francesco Castagna.
Hi, well, I'm Francesco Castagna from the University of Manchester, UK. And I was, sorry, maybe you can see me now. Was interested in, I mean, so far, I think I've understood that the main trigger for the interferon response is the genomic material of viruses, right?
Mm-hmm.
Yeah, but then you said that they found that even protein aggregates, prion-like compounds can be able to trigger this interferon response. Is that correct?
Mm-hmm, mm-hmm.
Yeah.
No, sorry. It wasn't a prion-like aggregate. It was actually MAVS itself.
Okay.
Together with the Rigi that is forming these aggregates, and the aggregates look a little bit like kind of structures you get in prion disease. I don't know. I think they were just going for like a sexy title or something like that. But
okay.
no, these clearly are like aggregate structures, yeah, of multiple units of MAVS protein interfacing with one another, coupled with the multimerization of Rigi itself. So. Okay.
Yeah, because my question was, are you aware of any other protein aggregates from any other source able to trigger the interferon response? Because I work on the immune response, the innate immune response to protein aggregates and therapeutic protein aggregates. That's why I was interested in that.
Well, but you must be able to tell me that.
Yeah, I mean, just started my work, and I didn't come across to interferon response to protein aggregates so far. Yeah. So I was.
Yeah, I'm not aware of it, but I, honestly would not be surprised if there were kind of intracellular structures and aggregates that in some way could trigger an antiviral response through activation of NF-kappa B or activation of other toll-like receptor structures, for example. So I'm focusing very much on the Rig and Sting pathways. Those are very important in sort of cytosolic response to viruses. But in fact, there may be situations where you also can get aggregates that will affect some of the TLR components, or indirectly other sensors. So.
Okay. Thank you very much.
It'll be interesting to look. Be interested to hear from you on that.
All right.
Fabiola Vacca has a question. Please, Fabiola.
Hi, can you hear me?
Yes.
Yes.
Hi, I'm Fabiola from Toscana Life Sciences, from the University of Siena. And I was, in terms of MAVS, as we were already speaking before, since they have those kind of prion-like properties, I was wondering if, since they can aggregate, is it possible for them to spontaneously also aggregate, leading to accidental activation in cells? Since, as we know, prions are also can lead to diseases and everything. Do you know of any case of MAVS that can spontaneously aggregate?
Yeah. That's a very interesting question. I have to say that relatively early on in the analysis of some of these pathways, we did a fair amount of confocal microscopy on MAVS and even on Rigi, but particularly MAVS and the mitochondria. We did not see inappropriate activation of MAVS, perhaps reflected as this aggregation. But once you had a virus infection, there were a whole range of different phenotypes of the cells that you could see by microscopy. including the mitochondria themselves fusing. And this is mediated by mitofusions and so on. So you would actually see the aggregation of the mitochondria. But I don't think that happens spontaneously. So I think that this is actually a fairly precise mechanism. You saw that there was the activation of Rig and then the dimerization of Rig and the oligomerization of that, as well as the oligomerization of MAV. So I think it's an extremely closely controlled mechanism.
Okay, thank you.
Thank you.
Question by Eleonora Sala.
Hi, I'm from San Raffaele University. I have just a general question. You talked about activators and repressors of STING, but is STING also characterized by a suppression mediated by epigenetic signaling that in turn block the cytokine production?
Good question. I'm going to switch off your your video feed. No, that's a good question. I don't. Yes. Now I'm thinking about it. Yes, there are examples of STING actually being repressed. The path, and it's an epigenetic mechanism. Don't ask me more details than that right now. But yes, there are examples of the pathway being shut down. And this often happens, not often, but it happens in some cancer cell models, so that these cells are no longer responsive to danger signals or extracellular signals. And the result is you might expect that to occur actually, because STING would normally trigger not just an antiviral response, an inflammatory response, a cell death response that would kill the cells. In a tumor model situation, in some cases you may need to repress that pathway so that the cells maintain the ability to grow. So it is a target. It has been shown to be inhibited by epigenetic mechanisms.
Okay, thank you. I have a question by Giorgia Manni, who asks me to ask the question because she has problems with the microphone.
Okay.
So I saw in your slide that IDO1 is induced after virus infection, and we know that IDO1 activation induces immune tolerance. Mm-hmm. Is it possible to hypothesize that IDO1 inhibitors as antiviral drugs?
Yes, it's possible, but yes, it's possible, I think. But then you would be really restricting a very specific mechanism. And honestly, I don't know of whether anyone has tested IDO inhibitors for in terms of their potential to modulate antiviral response. But remember, of course, that there are, of course, going to be a lot of different genes that are activated and pathways that are stimulated by the response. So I don't think it's been well characterized. Yeah.
Okay, and another question by Andrea Uva, who also has problems with the microphone. Is it possible for STING to be constitutively activated in cells, for example, mutations? And what kind of medical features could be linked to this activation? Mm-hmm. For example, vasculitis, he says.
Yes, yes, and yes. Constitutive activation of STING and the mechanisms can be fairly complex, but it can lead to chronic inflammation and autoimmunity problems. There are a couple of diseases that are in fact associated with chronic STING, Cardes-Gutierrez syndrome, and SAVI, which is severe acute vasculitis in children. So these are actually genetic mutations that lead to constitutive activity, and they have a very serious autoimmune phenotype associated with them. There may be more. It just hasn't been investigated as yet.
Okay, thank you. I think we're done with the questions for now. Yeah.
All right, let me just, okay. So I want to turn now, I want to turn and I'm going to talk about a specific virus. I'm going to talk about dengue virus. I know in this world these days, it's very hard to think about other viruses, but the dengue is a very interesting one, I think, to consider in this regard. And it's something that we have been interested in understanding molecular mechanisms as well. So dengue virus. So it is a member of the flaviviruses. It's an RNA virus. And it is spread by the bite of a mosquito. It's the most common arthropod-borne virus in the world. The mosquito is the Aedes aegypti by and large. About 2 billion to 3 billion people are at risk for dengue virus infection, particularly in tropical and subtropical regions. And that risk is increasing over time because of, as you can imagine, climate change. It is estimated that there are 300 million to 400 million infections per year. This can result in millions of hospitalizations per year and annually anywhere from 25 to 50,000 deaths. The primary infection with dengue is often considered a self-limiting acute infection. Anybody who has had dengue will tell you about break-bone fever, which is the way they describe the fever that ensues after the infection. It's severe enough that they feel like their bones are going to break. Secondary infections, secondary heterologous infections, can be even more dangerous. There are 4 different strains of dengue virus, and the secondary infection can often lead to a more severe immunopathogenic disease with a risk of hemorrhagic fever or a septic shock phenotype. At this stage, there is no effective antiviral treatment, no effective vaccine. I look at this slide and I sometimes find it a little discouraging that here we are in the midst of a very serious pandemic, and yet I don't know if there are too many people that are aware that dengue is actually such a serious human pathogen in the world. Largely, let me just go on here, largely because the infection is occurring in the tropical regions, in South America, in Southeast Asia, and in India, of course, through Central Africa and so on. And so often because these are in poor tropical countries, really has not received perhaps the same attention as we are seeing now with coronavirus. And I think there are lessons to be learned here. And this is really quite an interesting immunopathogenesis. These these isotherms, it's a 10-degree isotherm. So most of the areas at risk for dengue are in the tropical, subtropical regions, and these isotherms are expanding at a significant rate because of global warming. So far at home in Canada, we're safe, but we shall see. It's also considered an area where there is not very much dengue. On the other hand, there have been chikungunya outbreaks in Italy and some SIICA. That probably means that dengue is also prevalent to some extent because it is the same mosquito that carries these three different pathogens. The virus itself, I'll just go through this very quickly. as I mentioned already, there are 4 different strains of dengue, 4 different serotypes. The virus itself is a positive strand, single-stranded RNA. It is recognized and responded to by the Rigi pathway. And the initial production of virus actually results in a long genomic polyprotein. This occurs in association, or the replication occurs in association with the endoplasmic reticulum. And then using a viral protease called NS3 protease, shown here, there's specific cleavages of this polyprotein to generate the mature proteins of the virus. 3 proteins, capsid, matrix, and envelope, this is the surface glycoprotein, are encoded by the N-terminal end of the polyprotein. And then there are 5 other non-structural proteins. And these non-structural proteins are part of the mechanism by which the virus gains access to the, or control over host immune response. I mentioned earlier pathophysiology. I mentioned earlier that secondary infection with dengue can lead to a very severe disease. Some of the characteristics of severe dengue infection, including hemorrhagic fever and so on, are a cytokine storm, massive release of cytokines that induce endothelial changes in morphology of endothelial cells. This can lead to vascular permeability, so the capillaries become leaky, increase the expression of adhesion molecules, and as implied by cytokine storm, a very large production of cytokines, chemokines, including interleukin-1, IL-6, and several others. The consequences of this cytokine storm and the loss of vascular integrity, you end up with the plasma leakage, edema, hypovolemic shock, and so on. So these are the immunopathogenic consequences that are associated with severe dengue virus infection. And studies over the last number of decades have identified a number of immunological mechanisms that contribute to this severe dengue. They can include antibody-dependent enhancement, activation of complement, which, as you can imagine, can be deleterious in vascular tissues, oxidative stress mechanisms where you produce excess amounts of reactive oxygen species, and also selective T-cell clonal expansion. The risk of getting a severe dengue infection is most closely associated with this phenomena of antibody dependent enhancement. And it is seen, first described actually in regard to dengue virus infection. And my next slide is a simple schematic of that. If an individual is infected with serotype 1 of dengue, as I said, they could have an inapparent infection, they could have a severe acute infection that they recover from, but of course they will generate antibodies, and those antibodies will neutralize serotype 1. However, if that individual is infected with a second serotype of dengue, the heterotypic antibodies from the previous infection, the antibodies that are protective against the first strain, first serotype, are not necessarily neutralizing for the second infection. The result is they may have partial neutralization, or they may be present at low concentration. The result is that these virus antibody complexes are floating around in blood and in tissue, and now have the capacity through the C-terminal antibody receptor to bind to the surface of adjacent cells, cells that normally wouldn't be infected by the virus, and that would be occurring through the Fc gamma receptor, now leading to uptake of virus by these other cells, ultimately leading to increased virus replication and spread of the virus through the body with increased virus load, and also the activation of this massive cytokine storm that contributes to disease activation. So increased virus load certainly can be one of the factors. The other immunopathogenic mechanisms that I've just mentioned are also playing very much into this. But antibody-dependent enhancement appears to be, in the case of dengue, the strongest correlate of severe disease pathogenesis. I want to just divert for a minute and say, as you can imagine, this is a severe viral disease that has been also a target for antibody development for almost 20 years now. And the first live attenuated dengue vaccine called Chimeravax was generated by Sanofi Pasteur. They used a different vaccine strategy, or they used a vaccine strategy. There are several different strategies that are being pursued by pharmaceutical companies, but they used a Chimeravax strategy that actually used the yellow fever 17D genome, yellow fever vaccine. The yellow fever vaccine, which uses this 17D antigens to generate antibody response, is really one of the most effective vaccines known to man. It will completely decrease levels of yellow fever, triggered also by a flavivirus. So the strategy of the Chimeravax technology was to remove the surface glycoprotein, the E protein, and one of the precursor proteins from yellow fever, and replace it with the surface receptor structure of the 4 different serotypes of dengue. Resulting now in a chimeric yellow fever backbone expressing these surface proteins of dengue. That's then transfected into cells. Virus is grown in culture, and the resulting virus now is essentially, it looks like a yellow fever vaccine, but it's now expressing the dengue virus glycoproteins on its surface. And that has been used as a vaccine candidate. It has been through phase 1, phase 2, phase 3 trials, and generally a good vaccine. It shows 57% overall efficacy. It reduced, in clinical trials, reduced hospitalization by 80%, and also reduced dengue hemorrhagic fever by up to 90%. So it has been released, a fairly limited release in select populations, in select at-risk populations. One of the trials that was initiated was called Dengvaxia program. It was initiated in the Philippines, and it was a school-based vaccination program. The initiated April in 2016, and it was 700,000 immunizations of school children. By November of 2017, the program was suspended, and it was suspended because there had been the death of 64 children with no prior history of dengue virus infection. So the rationale was excellent, using a yellow fever vaccine strategy. The clinical trials were not perfect, but were good. and it was expanded into a school-based vaccination program, and yet was suspended with because of death of young children for reasons that really have not been well characterized. It was known that one of the problems with this vaccine is that it was not particularly effective against one of the serotypes. I believe serotype 4, but don't quote me on that. And so there was some concern that it may not have the kind of broad range protection that would be required. And so this is, and I think the outcome here, you know, it of course raises a number of questions about vaccination and about the acceptable risk of vaccination. This is essentially a 1 in 10,000 risk. You know, what is acceptable risk? Is it 1 in 100,000? Is it 1 in a million? Is it, you know, infinite? So I mean, this raises a number of questions about vaccine development. And this is why, of course, in the age of coronavirus, we often are talking about, you know, trying to develop a vaccine, but that the development of that vaccine is really going to take time until these tests are thoroughly completed. So I digress a little bit on that because I think it's an important aspect of what has gone on with dengue virus pathogenesis. It's not for wanting to get a vaccine that they have pushed forward. There's just one slide from an old review article that we made. Dengue infection disrupts antiviral immunity. And here you see the non-structural proteins 2, 4, 5, NS2A, NS2B3. They also have a very powerful mechanism to disrupt early interferon signaling at multiple levels. Not only in the initial sensing and response to virus infection, okay, we had dengue and we had Zika in that review, but also in regard to the interferon pathway and JAK-STAT stimulation of the interferon stimulated genes. Okay, I want to discuss just in a little bit of detail one mechanism of dengue immune evasion. Dengue virus has been shown rather convincingly to cleave sting, and it uses its viral protease, the NS2B3 complex. That's the protease that cleaves the polyprotein of dengue. It also turns out to have some cellular targets, and one of the cellular targets is the human sting protein. And I'm just going to show you a bit of data here. Don't get, let me just, there we go. So here you can see an immunoblot of human sting. This is coming out of myeloid dendritic cells. And here you can see a cleavage product of sting. And this cleavage of sting is being generated by the expression of the NS2B3 protease complex of dengue. That protease will also cleave the NS3 down to that complex down to a smaller form. So you can see it's still working on the viral peptide, but it's also cleaving sting. If you make a mutant of the protease, where you inactivate the protease cleavage site, you now see that you don't generate any cleavage product of sting, and you also don't get any cleavage of the precursor of that complex. Situation's a little bit different in murine cells, and I don't think you can see it, but murine cells don't undergo this specific cleavage. Why? Because the particular site that has been defined for cleavage of sting is not conserved in the murine sting. So in fact, you get, it's human cell specific, or human sting specific for this cleavage event. Okay, so an RNA virus generates a big RNA. It's recognized by the Rigi and is used here to stimulate antiviral responses against it are coming through Rigi. So why cleave sting? That was sort of an interesting, perplexing question. But in fact, what the answer is relatively straightforward. I perhaps have alluded to it already. I will just show one slide about that. And that is there's a significant crosstalk between these two pathways. I'm just going to show it here. This is part of the data. If you infect now lung epithelial cells with, in this case, Sendai virus, you can see that you not only trigger an antiviral response, but you activate Rig and you upregulate Rigi very nicely. If you work in a cell line, this is a human hepatocyte cell line. It also will get infected. It will upregulate ISG, interferon stimulated genes, Rigi itself, and sting. Here is a version of that cell line that has a point mutation in Rigi, so it doesn't do any Rigi signaling, and you get no upregulation of sting in this case. And there are several other examples here. If you mutate, if you knock down and silence the MAVS protein, of course, central to Rigi pathway, you again don't get any sting activation. So I'm just going to go ahead, and this is, okay. So this is now actually done in vivo, where animals that are knocked out for MAVS likewise don't respond to 5' triphosphate RNA by upregulation of sting. And this occurs in different tissues as well. So this seems to indicate, and I think it's illustrated here schematically in the review article from Alessandra, that on the one hand you have this is a recapitulation of the Rigi pathway, Rig, MAVS, the whole triggering of the antiviral response. The other arm here of the sting pathway, sting being stimulated through C-gas again. But there also is evidence for physical interaction between the adapter molecules, sting and MAVS, but also many of these proteins and adapters in both of these pathways are actually interferon stimulated genes. So this is part of the mechanism for amplifying the antiviral response. Now, why would sting be a target here? Well, I think it's a target because not only does dengue recognize sting as being a critical antiviral protein, but also in the cells that are infected with dengue, there is often the release of mitochondrial DNA, and that will trigger the sting pathway. So this seems to be a mechanism, again, somewhat ingenious, to knock out the sting pathway so that these cells essentially become insensitive to DNA damage, as well as the triggering of the Rig pathway. Okay, so I have not really talked about it too much, but there are ways to synthetically modulate these pathways. One is the generation of 5' triphosphate RNAs. We've generated one previously that actually has a very high capacity to trigger the Rigi pathway and also stimulate sting. And then CGAMP is also, as you've heard, an important activator of the C-gas pathway and C-gas sting pathway, and is also one of these targets for pharmaceutical development along this line. So last part, I want to just very briefly go through dendritic cells and dengue virus infection. We were able to show, or a number of groups have shown, that the skin resident dendritic cells, Langerhans cells, are often the initial target for infection by dengue. The mosquito, just picture the mosquito coming in and biting your arm. So the immediate cells that they encounter are dendritic cells and Langerhans cells. And you get a very rapid inflammatory response, antiviral response. You get, of course, this interference with STAT signaling and so on. And this is quite a crucial interaction for both the replication of virus and for ensuing evolution of disease severity. This is a slide that I like this slide because it illustrates in a very clear way that if you take monocyte cells from an individual, they are largely resistant to infection. It's as they differentiate to dendritic cells that they become more and more sensitive to dengue virus infection, up to the point where you can get almost 80% of these cells being infected. And that is reflecting increased maturation and also increased expression of surface receptors on the dendritic cell surface, particularly the DC sign molecule, which serves as a receptor. If we go back to, you remember some of these gene networks that I showed you earlier, again, in dengue virus infected dendritic cells, you get activation of inflammatory cytokines, the IRFs and so on, NF-kappa B signaling and TNF. But you also get the activation of a network of genes, which I did not know very much about. And this is a network of genes called the NRF2 genes, antioxidant response genes. And this is a whole network of a couple hundred genes that play a role in redox homeostasis and protection against oxidative stress. And these are being regulated by this NRF2 molecule. It's a basic leucine zipper transcription factor, and it binds to a specific antioxidant response element that is present in, as I say, hundreds of genes that are involved in glutathione synthesis, reactive oxygen species elimination, drug excretion, NADPH activation, and so on. NRF2, similar to some of the patterns that you've seen earlier, NRF2 is localized in the cytoplasm. It is partnered with a protein called KEAP, and KEAP actually attracts ubiquitin proteasome complex proteins that cause the ubiquitination and degradation of NRF2. In response to oxidative stress, virus infection, electrophiles, DNA damage, and so on, there are a number of activators that will lead to the modification of KEAP that releases NRF2. Finally, it says, finally, I'm away from that KEAP, because it stabilizes NRF2 to now localize to the nucleus, and then together with other small proteins lead to activation of these antioxidant response elements and the triggering of all of these genes. So is there a link between this oxidative stress response and dengue and the antiviral inflammatory responses? The answer is yes. NRF2 and this antioxidant response, they reduce both antiviral activity and cell death responses to dengue virus. If you silence NRF2 by RNA interference, or we've done it also with the knocking out, you increase dengue associated immune and apoptotic responses. So it has some sort of a role in modulating that early innate response that I've talked about. This is some data that confirms that. There has been what I think is a very interesting breakthrough relationship in terms of metabolism, NRF2, and the antiviral response. And it comes from a couple of really great groups. The first observation was made by Mike Diamond, who's at Washington University, St. Louis. And he discussed and described that a Krebs cycle metabolite was able to generate macrophage remodeling and regulation of inflammation, basically suppression of inflammation. And as we had wondered, we knew that NRF2 and the generation of the antioxidant response also had an effect on limiting innate antiviral responses and cell death in dengue infection. And the next part of this story came from Luke O'Neill's group at Trinity College in Dublin. He has really been a leader in understanding the relationship between metabolism and immunity. Here is the Krebs cycle. I won't go into a lot of details about metabolic pathways, but as a consequence of, in his case, he did treatment with lipopolysaccharide, so occurring through TLR4. We've done some experiments using viruses, including dengue virus, to look at this question. And essentially, this molecule itaconate is a byproduct of the Krebs cycle. It is a byproduct of citric acid metabolism. It takes cis-aconitate and converts it to this byproduct itaconate. And itaconate actually is able to suppress inflammation through the induction of our friend NRF2. So, and how is it doing it? Well, it actually creates a new type of alkylation on the KEAP protein, so releasing NRF2, and leads to the upregulation of multiple NRF2 responsive genes. So itaconate is an anti-inflammatory metabolite. It activates NRF2 via alkylation of KEAP. This was in a very nice paper in Nature recently. And it also, as a consequence of triggering NRF2, suppresses antiviral responses. This is some studies that were performed by David Lelanier, who is now in Aarhus. So here is now a use of itaconate. It's a very simple experiment. We take that antioxidant response element that is present in the NRF2 responsive genes, increasing amounts of itaconate for oxyitaconate here will increase the expression of the ARE and increase expression of NRF2. There's another compound that has been used extensively, sulforaphane. This is an isothiocyanate molecule, and it also is a very powerful activator of NRF2 through the same mechanism. Essentially, it modifies KEAP. So what's it do? Here is a simple experiment that gives you an answer. Here are cells that have been stimulated by cGAMP. So they stimulate the STING pathway and they produce lots of interferon. On the other hand, itaconate appears to completely suppress the production of type 1 interferon. And now if you look at some of these Western blots of these cells, you can see that GAMP, nice activation of STAT, interferon stimulated genes, phosphorylation events. Boy, you've probably heard too much about those, TBK, IRF3, and so on. But in the presence of itaconate or sulforaphane, there's a very powerful repression of the cGAS-STING pathway. So not only the inflammatory pathway, but also the antiviral pathway can be controlled by this metabolite and by this small molecule isothiocyanate, which actually has moved into clinical trials. Well, just want to end now with a sort of what I think is a rather complex mechanism of interrelationship between metabolic reprogramming. So cells that get treated with LPS or cells that get infected with the virus, they upregulate glycolysis, they switch from oxidative phosphorylation to Warburg glycolysis. And the citric acid, the Krebs cycle is active in these cells. A molecule called immune response gene 1 or cis-aconitate decarboxylase is responsible for converting citrate to itaconate. Itaconate will stimulate this NRF2 network, and in turn, these will have an impact on inflammation and the interferon signaling pathway. I say it's complex because NRF2 has also been associated with a number of different regulators, including hemoxygenase, which seems to have some antiviral activity. And then this cis-aconitate decarboxylase, IRG1, actually learned about it first as interferon responsive gene, because in fact, this is an enzymatic activity that is being stimulated by interferon. So you have glycolysis affecting host metabolism. You have the induction of this particular enzyme leading to a taconate, negative regulation, but then there's also a loop that kind of goes back and stimulates immune metabolism. So it's a complex network, but a very interesting one that links metabolism to antiviral responses. So my conclusions: dengue. I guess we would have to call it an emerged global pathogen that infects dendritic cells. It's coming from the bite of the mosquito, Aedes aegypti. It results in a disease that ranges from inapparent infection to life-threatening immunopathogenic hemorrhagic fever. Dengue can trigger antiviral, inflammatory, stress-related pathways, as well as regulation of reactive oxygen species. And then this link between NRF2 and metabolic signaling can control the antiviral response and links actually metabolism to antiviral response. Very complex switch involved here, just between metabolism and innate immunity. Likewise, as we've discussed with disease pathogenesis, a very complex link between dengue virus and the more severe disease. And as we are seeing right here now in relation to the COVID pandemic, also a very complex and poorly understood switch between what happens in patients who recover from the disease and those who go on to develop life-threatening conditions. And this is obviously going to be multifactorial, but very, very intimately related to immunopathogenesis. So I'll end with NRF2 network shown here. NRF2, it's really at the interface between metabolism and antiviral immunity. Here are the pathways, here are some of the signaling molecules. And as NRF2 activity goes up, innate immunity goes down, and this probably will oscillate depending on the state of metabolism in the cells. Okay, I want to thank a number of people from my lab, past and present, who have contributed to this and other work. Here are some of the very extended family. Certainly I thank the funding agencies that have kept the projects alone, to have kept the projects funded and going along. And finally, I just want to thank you for your attention. And in this day and age, I guess we have to say, stay safe. My last statement, okay, that's my question. It's kind of okay, maybe it's an easy question, maybe it's a very complex question. I don't know how easy it's going to be to answer this question in 100 words, but are there lessons to be learned from dengue immunopathogenesis that can be applied to the current COVID-19 pandemic with regard to therapeutics, vaccine development, and even pathogenesis? So I will be very interested in your answers. Thanks very much.
Thank you, John, for this wonderful lecture, and actually very timely. And I think we are open for the last leg of lesson, question, sorry.
Yeah, are there any questions from the students on Zoom or also from YouTube?
Has everybody turned off?
No, there are still almost 200 people are still on.
Okay.
But maybe they asked all their questions before.
Maybe, okay.
Yeah.
So I have a question for the students. Actually, do you have, or have you had lectures on dengue virus immunopathogenesis?
Okay, waiting for them to answer. Yes, somebody says never.
Never.
Francesca, no, never. Nobody.
Yeah.
Yeah.
Really, I find it fascinating because, of course, as we go through the reality of this pandemic, I find that there are many related and unanswered questions that are also coming from the whole issue of dengue immunopathogenesis.
Yeah, so there's a question from Andrea. He also cannot use the microphone, so I'll read it for him. Could a vaccine that ideally induces a strong cytotoxic response help with or limit the risk of eventually occurring ADE?
A vaccine that would strong.
The risk of eventually occurring ADE, meaning antibody
dependent enhancement would be, yeah. I mean, so you're talking about a vaccine or a therapeutic that would essentially kill infected cells so that they are unable to generate a antibody-dependent enhancement effect, or yeah.
Yes, I don't know, Andrea. If you want to specify more, please write on chat.
Okay, no, it was it's an interesting idea. I think I think at this point, therapeutics have not been very helpful. And so along, as we also encounter now, the long process of developing an antibody is the preferred way to do it. In the case of dengue, of course, you could be considering whether you want to generate a predominantly antibody-dependent vaccine, or whether you also want to enter into a T-cell-based vaccine that would then target and kill infected cells.
Okay, thanks. Question from Riccardo Capecchi. Riccardo, can you hear us, please?
Can you hear me?
Yes.
Hi. Thank you for your presentation, to your lesson. Very interesting. Thank you. I just thinking about the problem of the low affinity with antibody response in dengue, with the problem at the second infection that could be at higher risk of worse income. And I just think that this model could be a risk for some type of vaccination, of course.
You got it. Yes, I can stop you there and say you are quite correct. There are many people who have studied dengue a lot longer than I have, who questioned whether it was advisable to generate an antibody-based vaccine, because you would have to generate a vaccine in which all of the antibodies being generated against the 4 serotypes would be equally effective in eliminating the virus, because of the risk of antibody-dependent enhancement with secondary infection. And I should say that some of the regions of the world, Brazil, for example, Southeast Asia, it's no longer a matter of, you know, in one season, these outbreaks, epidemics, if you will, they occur seasonally, and they occur during the hot, humid weather when the mosquito is able to multiply very easily. And these outbreaks now, in one season, it used to be associated with a particular serotype. Now, all serotypes seem to be prevalent throughout the world, and probably the increasing prevalence of mosquitoes and the serotypes are what are kind of driving the increased severity of the disease on a seasonal basis. And so, yes, the generation of antibodies with a vaccine really has to take into consideration the ability to completely neutralize all 4 serotypes of the virus. It is the risk factor. I'm sure several people probably several dengue vaccinologists pointed to the problems in the Philippines and want to blame it on just that fact. It might be a good deal more complex than that, but that is absolutely a risk. Yeah.
Thank you so much.
Thank you. Francesca Millon from SIICA Junior Faculty has a question.
Good afternoon, Professor, and thanks for your talk. I had a question about the chimeric vaccine for dengue infection. So maybe I missed the point, but what was wrong with the vaccine? I mean, why did they, I mean, we had this 64 children that unfortunately had problems with the vaccine, but did they understand why?
No, that's, I think the bottom line is it's not understood. In a sense, it is, how to say it? In a sense, it is, I think, discouraging that it happened in the Philippines, because I think the response of the current government in the Philippines was to arrest people involved with the vaccination program. It's not clear. The origins of this problem are not clear. But as I said, there was one serotype of dengue that was not as efficiently neutralized as the other 3. And potentially that is related to the issue with the vaccine efficacy. I mean, I think that is sort of one of the first things that could be considered. The other possibility is that there may be underlying issues with the children that were not previously recognized. So I think those are a couple of things. You know, I don't know if I'm sympathetic to this or not, but the companies, you know, they probably spent $1 billion to develop the vaccine through the clinical trials and so on. And based on their best estimates, based on the phase 3 trials, these were effective and safe vaccines. But clearly there's a limitation. And I think perhaps it may go back to the fact that one serotype of the virus was not completely neutralized.
Thank you.
Thank you.
Okay. A question from Andrea Uva. Again, his microphone doesn't work. Can we think to use I cannot say this, itaconazole, yeah, in interferonopathies?
Sorry, in
interferonopathies. Interferonopathies. Sorry,
yeah, this is you can guess this is a very hot area right now. Trying to look at the possibility of using itaconate as a regulator of inflammation or autoinflammatory conditions. You want to suppress the antiviral response. Yes, we are looking at that. Many groups are looking at that. In our case, we use sulforaphane to mimic what we now know to be the itaconate effect, and it's a very good suppressor of antiviral immunity. Sulforaphane is already in phase 2 clinical trials in cancer therapy. I don't know the details of that. So there is a lot of interest in trying to modulate the immune response via metabolic agents that would have an impact on viral infections and cancer.
Okay, thank you. And Alessia Metrano from Naples is asking, could ADE explain the pathogenicity of severe COVID-19?
It's a possibility. I think that I have heard, I have not seen that there may be a second strain of COVID-19, one that is more pathogenic, one that is less pathogenic. But in fact, we are looking at this point at a, so far at what I think is an infection with a single agent. Could you generate antibodies that are, non-neutralizing and therefore contributing to pathogenesis by another mechanism? Yes, absolutely. So I think that these are all things that need to be looked at. Any of you who have seen your bosses lately, you know that we are all frantically writing research proposals to address just these sorts of issues, the nature of inflammation, why the virus seems to be a very low-level chronic infection for a while, and then in some patients just takes off after a week or so. So yes, I mean, this and many other questions need to be investigated thoroughly, but generation of ineffective antibodies is certainly one of the ways that the pathogenesis could be increased.
Okay, thank you. And a question from a YouTube follower. Are there any data?
Sorry, I'm laughing because I'm hoping that my daughter in Canada is not watching, because she said she was going to interrupt with some stupid questions. Okay, sorry, can you repeat?
To the other one, it was about AD mechanism in SARS-CoV-2 infection pathology. So I think you already.
Yes. Yes, I mean, I think that's kind of the best response that I have at this point. That yes, there could well be an impact of antibodies on the immune response. Okay, reservoir animals. Yeah, this is really an interesting question. How do they cope with it? I don't think there is very much that is known about this, but at this point, but clearly these animals probably have sufficient antibody or T-cell response to the virus that it remains kind of a low-level infection in these animals, but nonetheless transmissible. It would be very interesting to investigate these aspects of how viruses are maintained in reservoirs like in the bats and so on. That being said, it is not easy to investigate this, because in our immunological world, we need a whole lot of different reagents to be able to investigate, the human immune system or the murine immune system. And the first thing that would be necessary here is to generate an entire network of reagents that would be amenable to analysis in bats. But I think it is really it's very an important question. This also, the issue is relevant for other infections, and most notably Ebola. How do these animals tolerate the virus?
Okay, thank you. There are no other questions, I guess. So thank you very much for your lecture, and thank you to all the students for their participation. I think we all enjoyed this.
Thank you very much. It was fun.
Thank you.
Take care.
Thanks.
Thank you very much.
So Silvia.
Yes, thank you. If you can hear me, I say again, thank you, John. And now we move to the next session of the school. Before going to the group briefing, I would like to say goodbye to our YouTube users. So the live streaming on YouTube is closing now in this moment. Please follow our Facebook page to stay updated. So next webinar will be on Tuesday at 3 o'clock